Hello, today we will discuss about the antibodies, immunoglobulin. So what is that? And we will also understand the classification of the five immunoglobulin classes. So let's begin to understand. Here is the person which that contain a blood circulatory system and lymphatic system. Lymphatic system which that is important for the natural naive T cell and naive B cell reside on the lymph node of the lymphatic system, and this is the blood circulatory system. So the blood circulatory system is very important for their many lymphocytes. So the lymphocytes, because it is the part of lymphatic system which produce the lymphocytes, and the lymphocyte is basically the produced from the stem cell in the bone. So ultimately go and immature T cells and B cell will reside in the lymphatic system. While here is a circulatory system, you can think about that. This is the blood circulatory system, and this is the naive B cell and naive T cell, which that is present in the lymphatic system. Basically, in the thymus, so the T cell are present in the thymus mostly, and B cell also. So, B cell will educate in the thymus gland and will proliferate to target the antibody producing mechanism to produce antibody for the adaptive immune response. So let's begin to understand. Here is the antigen presenting cell which that is contain toll like receptor. This toll like receptor, when virus will be contact with the toll like receptor, it will give the signal inside to produce the chemokines, cytokines, etc. And that chemokine and cytokine will recruit the T cell and T cell and activate the T cell. After the T cell, naive T cell will proliferate into the CD4, CD8 and T regulatory cell. Anyhow, after this basically is the CD4 is used for the T helper cell, T helper, and help and recognize the antigen and give the produce the interleukin for recruitment of the naive B cell, and also go into the lymphatic system for activation of the naive B cell. And after this activation of the naive B cell with a specific antigen will show with the T cell antigen presenting cell as a antigen presenting cell to the antibody of the B naive B cell. After the naive B cell will recognize the antigen, so it will proliferate with a specific B memory cell and plasma cell. Plasma cell produce antibody in the infection time, while the on the other hand, the B memory cell will produce antibody throughout the life for further again infection clearance. So that is why it is known as the B memory cell. So after this basically the here is we will discuss about the naive B cell will proliferate on the basis of their specific antigen, which that is come from the toll like receptor of the dendritic cell. After this recognition, so produce the antibodies from the plasma cell and the time of infection. And opsonization of pathogen will be occur when collapse the antibody around the antigen. So the antibodies is also known as immunoglobulin. Let's begin to understand the major things. So first of all, here is the opsonization of pathogen will be occur, and in this way, the neutrophil and mast cell basically used for the endocytosis after the neutrophil, and neutrophil will be endocytosed to produce lysozyme and hydrolytic enzyme and other enzyme which that will be neutralize this antibodies containing antigen. So after this neutralization, the macrophage will be eat the big eater. It is also known as the macro mean bigger, phage mean eater. So that big eater will eat and clear the pathogen. So immunity can be two type, three type, but here we will understand just two type: innate immunity and adaptive immunity. Adaptive innate immunity contain macrophage, dendritic cell, mast cell, neutrophil, and basophil, or any other also. But here the adaptive immunity produce the antibodies through B cell and T cell activation, can be passive immunity or active immunity. So the passive immunity is the external antibody, artificial media and vaccination, and self-stimulate is the active. So here is I am drawing the antibodies, the immunoglobulin, the bigger picture for understanding. So it's contain two chains, two large chain and two smaller chain, light chain and heavy chain. So it is here is the contain carbohydrate with a CH2 heavy chain, while the light chain are present in the periphery of the hemoglobin in the antibodies. So this is the disulfide bond. That's disulfide bond is very important for the retaining of their structure. While the antigen binding site is here, which that is used for the binding of the antigen. While the downside is that this is the structure is Y shape, and here is the heavy chain which that is interconnected through a polypeptide. So it is also made up of uh, proteins. That is why it is known as the immunoglobulin, because it is the globular protein. And this is the heavy H chain, and this is the hinge region. And remember about that, this is the contain, there are many types. So here is the VH and CH1, CH2 and CH3. This is the heavy chain, and while the light chain contain VL site and CL light chain, because that is why it's known as a light chain, while heavy denoted by H. So the effector region, the FC portion, are present in the downside. So let's begin to understand about that. What is the FC portion is used for different purposes we will discuss here. So the 5 immunoglobulin IG classes we will discuss here. So that is 5 type can be IgM pentamer formation, IgM, IgG monomer, and secretory IgG dimer, and IgE is the monomer, and IgD is the monomer. Both are monomer. So if you are drawing the diagram here, so here you can see this is the pentameric way, and it is you can see that is the antigen binding surface of the antibodies is double, with a 5 antibodies will bind with a 10. So on the other hand, here is a 2, and this is a Y shape basically, and this is the dimer. Dimeric is secretory component are present in which that is used for the binding, and it is bind together. While on the other hand, other is you can see in this diagram monomer. So let's begin to understand the heavy chain. Its heavy chain is basically heavy chain is uh, micro, and here the gamma we can donate, and alpha and epsilon and delta. While here is the number of antigen binding site is the 10. Otherwise, others are 2, and dimer were 4, and other 4. Molecular weight can be 9 in the form of Dalton. You can see in this diagram, and you can think about that. And the changes, the more is pentamer for pentamer. So here is the percentage of total antibody in serum is the 6% in the IgM pentamer, while the 80% more are present the IgG monomer. While the it can be cross the placenta antibodies, but this basically is that one antibody can cross one antibody, that is the IgG, and that is why it is present the mostly in the blood circulatory system. So the 80% containing IgG monomer. So the Fc portion binds to first of all here is the phagocytes IgG monomer for, and mast cell and basophil with the secretory component containing secretory IgG dimer. While let's begin to understand about the function. So the function is the basically main antibody of primary response, best at fixing complement monomer form of IgM, serves as the B cell receptor. So the B cell receptor serving as a B cell receptor when will monomer, while it is in the form of pentamer. So you can see. And on the other hand, the IgG monomer can be main blood antibody of secondary responses, neutralizes toxin and opsonization. So for opsonization, the IgG is very important. IgG monomer. So on the other hand, the secretory IgG dimer, which that is secreted into mucus, tears, and saliva, colostrum. Colostrum means the beginning after the birth. The mother will produce the colostrum, which that is contain antibody. And on the other hand, the IgE is the antibody of allergy and anti-parasitic activity. And the IgD is the last one, is the monomer, which that is the B cell receptor, used for the B cell receptor. So I hope you make sense about these things, and this was the overview of antibodies. So please make sure to subscribe, like, and share. Ask the question in the comment below.